Welcome, everyone. Thanks so much for joining us today. My name is Rachel Massey, and I'm the Senior Science and Policy Advisor with CHE, the Collaborative for Health and Environment. We're really glad you could all join us today, and we invite you to put a note in the chat introducing yourself if you'd like to. I'm going to start with a few bits of housekeeping. CHE is a member group of the EDC Strategies Partnership, which is bringing you today's webinar. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, a program of environmental health sciences, the Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. If you're joining by phone, you can download the slides from the webinar page on our website at healthandenvironment.org. For those who are online, we'll put that link in the chat now. We'll disable the chat during the presentation, but please do type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentation. If you'd like to activate closed captions, you can do that by clicking the button on the bottom of your screen. This webinar is being recorded, and it's scheduled to last for 45 minutes. With that, I will hand it over to today's moderator, Cheryl Patton, Director of the Biomonitoring Resource Center. Cheryl?
Yes, hi. Thank you very much for the introduction, Rachel. And today's talk is going to be about PCBs and neural development. And just a quick review, PCBs are a family of industrial chemicals produced in the US between 1929 and 1979, used primarily in electronic devices, but also used in plasticizers in many paints, plastics, and rubber products, numerous applications in industry and building construction. They're still around, they're not really produced, but they're still in many kinds of items in the household goods and construction facilities. They're present also in fish, meat, poultry, dairy products, and breast milk. So dietary intake is an important pathway of exposure. Breastfeeding infants are exposed to PCBs in the milk of their mothers with previous exposure, and they can transfer prenatally from mother to the fetus. Recent findings suggest that the presence of PCBs in indoor dust and indoor air may also constitute an exposure pathway for some portion of the population. But clearly, PCBs are part of most people's chemical body burden. I'm really pleased to introduce our speaker today, Dr. Tom Zoeller. He's a professor emeritus in the Department of Biology at the University of Massachusetts, Amherst. His research focuses on the effects of the thyroid, which is essential for normal brain development. His research examines the mechanisms by which thyroid hormone action can be disrupted by polychlorinated biphenyls, PCBs, who are structurally similar to thyroid hormone. PCBs can alter the circulating levels of thyroid hormones, which can affect neural development. So, Dr. Zoeller, can you please begin your slideshow?
Sure. Thank you. Thanks for the invitation to speak about something that I spent a fair amount of time working on. So first of all, I want to give kind of a preview of the main points that I want to make. And the first is that PCBs are a complex mixture, and populations are exposed much more than just PCBs. So I can imagine interactions both within congeners within this class, but also outside of the class too. The effect of PCBs, for example, in people with low iodine might be an important element. Some PCBs can activate cellular responses that we generally think of as detoxifying, like in the liver. But this can also bioactivate some congeners, and I'll show you some of the data for that. PCBs produce this heterogeneous effect on thyroid hormone action in the developing brain, and I think that's one of the puzzles that we have to figure out why this is. Some parts of the brain in animals treated with PCBs look normal. Other parts appear to be sensitive to an overabundance of thyroid hormone, and some other parts of the brain look like they're hypothyroid all at the same time. I think this is a disaster in terms of brain development and brain function in the adult. But I want to show you some of those data. And also, as we think about these data, that some of this is 20 years old or so, but it does give us some insight into the way we need to think about mixtures and also of new approach methodologies, these NAMs that are sophisticated tests for molecular initiating events. And so how we begin to think about the use of NAMs in understanding hazard and risk, I think, is going to be really important. So to start off, there are two big stories that tell us about thyroid hormone and development. One is frog metamorphosis. There's great work on that. The way in which deiodinases are controlled during metamorphosis explains, for example, why the tail is the last thing to come off, despite the fact that serum T4 in frogs is very high during the whole metamorphic period. Also, the issue of cretinism told us a lot about how sensitive brain development, but also somatic development is in humans. So really in all vertebrates, with some exceptions, thyroid hormone plays an essential role in development. So we're not special in terms of a species. We also now know that the fetal thyroid doesn't begin to work really until about the 12th week of pregnancy, but it doesn't make enough thyroid hormone for the fetus's needs until about week 20. Thyroid hormone receptors are expressed long before this, of course, and we now know that the fetus is dependent upon thyroid hormone of maternal origin. Now, there were a couple of reviews. This one was written by Sue Shantz and Deb Rice. Essentially, what they did is review the literature, epidemiological literature, showing in all these cases that cognitive function or neurobehavioral issues are being affected by prenatal PCBs mainly. So there's a lot of human evidence as well as animal evidence that PCBs can affect brain development and brain function later. We knew when we first started this, we knew that PCBs could affect thyroid hormone levels, but nobody had really looked at what happens to thyroid hormone action in the brain following this. And this was kind of my first foray into this area. Clearly, PCBs can produce thyroid hormone insufficiency in rat pups during the first month of postnatal life, and this is an important time for thyroid hormone action. But surprisingly, when we looked at a readout of thyroid hormone action in the brain, what we saw is the reverse of what we expected. We expected the expression of genes that are known to be directly regulated by thyroid hormone to be suppressed, but instead they were activated. What was more important here, or certainly as important, is that RC3, this gene that's regulated by thyroid hormone, is regulated by thyroid hormone here in the dentate gyrus and in the cortex, certain parts of the cortex, but not in Ammon's horn of the hippocampus. And so we saw the same thing with PCB exposure. Only in those brain areas known to be sensitive to thyroid hormone was this gene upregulated by PCBs, which suggested to us a thyroid hormone-like action. We also saw in the cerebellum that PCBs produce an effect that looked much more like hypothyroidism. If we just look in this external granule layer here, cells are born along this external layer and they migrate across based on thyroid hormone levels. So in an animal that's hypothyroid, this zone becomes enlarged because the cells are not migrating appropriately. And this is also seen somewhat with PCBs. So one of the kind of bottom lines here is that PCBs can affect thyroid hormone action in different brain areas in seemingly a different way. one of the things that I've thought about quite a bit is how this might happen. Thyroid hormone is unlike estrogens and androgens and glucocorticoids in that it doesn't percolate through membranes passively, but instead is transported directly across membranes, first of all, into the brain, but also into, in this case, T4 is taken up into astrocytes or tanocytes, converted to T3, and then transported to neurons. This is actually an important mechanism, and we know in humans an MCT8 defect produces severe neurological consequences. So in the rodent, the setup is a little bit different. There's some transporters, not exactly the same, but still thyroid hormone doesn't gain access to compartments or cells passively. PCBs could potentially affect this kind of transport, could affect metabolism in tissues, might also be different in different tissues. And we've got data to suggest that, actually, to show that. So there appears to be at least 3 separate issues that might explain the heterogeneous effect. When I say heterogeneous, I mean that, first of all, the effect of PCBs on thyroid hormone action in the brain is not predicted by the effect of PCBs on thyroid hormone levels in the blood. And if we only look at blood, we're not really going to get a good picture in terms of hazard and risk. So one thing seems to be that PCBs can activate liver to clear PCBs themselves, but they also, because they produce hydroxylation events, can also, they can reduce serum T4, but they can also produce some congeners that may act on the receptor itself. Low serum thyroid hormone can trigger different cellular compensatory responses in different brain regions, which might actually be playing a role here. So there's a number of things that PCBs can produce that affect the thyroid hormone system. So we have shown that a hydroxylated PCB congener can bind directly to the thyroid hormone receptor. It's not a high affinity effect, but there's still a very significant binding, physical binding to thyroid hormone receptor. At the same time, we also show that this hydroxylated congener can activate, in this case, it's the induction of a growth hormone messenger RNA in a cell line. These are GH3 cells. When we use a specific promoter, we can see a more robust effect. This is a different promoter here that has less of effect. This is a mutated DR4 construct that shows the abrogation of PCB effect as well as T3 effect on TR activation. So one of the things that bothers me about these data is that the effect of this single PCB congener is different depending upon the promoter that we're using. In addition, when we look at this, this isotherm here, showing that PCB can bind to the thyroid hormone receptor, we're using a solubilized thyroid hormone receptor. This is a type 2 receptor that binds to DNA in the ligand-independent state. So in the apo state, this receptor is binding to DNA, and that DNA response element is different in different kind of genes. The context is different. We know that that context can affect the structural relationship of the receptor itself. So we don't have a good picture, I don't think, in binding studies that are done with a solubilized receptor, but that's the way everybody has done it. I think this is a significant shortcoming. and may explain some of the reasons that this PCB congener can activate thyroid hormone receptor in a manner that seems to be construct dependent or DNA regulatory element dependent, which is also going to mean how different genes are differentially sensitive to this PCB congener. It took several papers and a number of years to produce this cartoon here. What we have found is that dioxin-like PCBs can activate the expression of CYP1A1, get an increase in messenger RNA, increase in protein and CYP1A1 action, enzymatic action, which can then hydroxylate non-dioxin-like PCBs. In our case, it's 105 and 118 that can bind to the thyroid hormone receptor and activate thyroid hormone receptor target genes. We've got quite a bit of data, both pharmacological as well as expression data to demonstrate that. And I'm happy to pass along papers that we have that show this. This kind of two-state or two-step mechanism here we wanted to explore in humans. Now, placentas were available to us from an epidemiological study in Canada, and we know from other studies that the placenta exhibits known thyroid hormone target genes. In addition, CYP1A1 is inducible, for example, by cigarette smoke. So that seemed like a system that we could explore. We've got a lot of data in this paper, but what we show is that CYP1A1 messenger RNA in these placentas was correlated with both placental lactogen and the placental growth hormone messenger RNA levels. If we regress the expression of those two genes, we see a very tight relationship. This is, I think we had about 162 placental samples. So it seems to me that this is a pretty robust response, pretty tight. And it indicates to me that these genes are both being regulated by a single factor, by a common factor, but the expression of those genes was not correlated with cord blood T4 that came from those births. So in short, within the class of PCBs, some congeners induce cellular responses that can interact with other congeners to make them more toxic from the point of view of the thyroid hormone system. Because there are so many other dioxin-like chemicals capable of inducing CYP1A1, it implies interactions between PCBs and non-PCB chemicals. For example, women who smoke would certainly be more susceptible to PCB levels in terms of thyroid hormone action. Now, we can't draw a direct line from the expression of these genes in the placenta to brain development in the fetus, but it certainly makes plausible the idea that these kind of mechanisms are also occurring in the fetus as well as in the neonate. So when I think about mixtures, this is a paper that came out recently from it's a collaboration between people at risk assessors in the Netherlands and Andreas Kortenkamp in London. They did a mixture risk assessment on lead, methylmercury, arsenic, fluoride, non-dioxin-like PCBs, and PBDEs. What they found was that the median exposure of these chemicals was exceeded the combined tolerable levels for IQ at this median exposure level. So half the people in 9 countries that were evaluated have a mixture that is affecting IQ at the time. This seems like and these are foodborne chemicals. I think this is an important thing that we need to pay attention to. Secondly, I would also point out that these are just a couple of studies that indicate that chemicals can, in this case, PCBs, can affect DNA methylation that correlates with serum PCB concentrations and child neurodevelopment. Part of that neurodevelopment is affected by thyroid hormone itself. So even in the case of thyroid hormone levels that are normal, PCBs can affect the epigenetic control of genes that are known to be sensitive to thyroid hormone and that are also implicated in adult social behavior, such as autistic spectrum. Now, we can talk a lot more about that, but I think the combination of mixtures, but also new approach methodologies that are powerful at identifying a hazard on a specific molecular initiating event is very different from looking in vivo, and we have to be really careful in how we interpret negative results, but also what we do with positive results. I'd also like to point out, this is a little bit off topic in the sense that this is a PCB 95 that has a very high affinity for an intracellular calcium receptor, and Isaac Pesa had kind of collaborated with a group to look at whether or not this affected the organization of the auditory cortex. I think that that this paper really speaks to the way in which perception can be altered developmentally by exposure to these kinds of chemicals. And I think we need to take these kind of observations, which are hard fought. This is a very difficult kind of analysis to perform. We need to take this very seriously. So with that, I'll go ahead and stop and hopefully open up for questions.
Okay, thank you very much for that excellent presentation. It's just stunning, all the different pathways that PCBs can cause harm in neurological development. It's just endless in terms of the interaction of mixtures and what can happen. All right, so I don't see any questions coming up right away, but I do have some questions myself, which is we know that PCBs are in ocean water and they are absorbing into microplastics that then, of course, are consumed by marine mammals, fish as well. And then there has been some conjecture that some of the behaviors of marine mammals when they, when there's a lot of them that manage to beach themselves someplace in a way that's going to kill them, is a result of some neurological damage that may have been caused by the fact that they are taking in PCBs as well, and there may be neurological effects in marine mammals. What do you think of this hypothesis? Is that something that you might see is possibly likely?
Well, so I think marine mammals, I don't know that they would have a bigger mixture of chemicals that they're exposed to. I'm sure they're exposed to heavy metals as well. So I kind of, this issue with the auditory cortex really bothers me, because what it means is that cell contact dependent developmental events are being disrupted. These kind of things, to me, will alter the way animals are going to respond and humans are going to respond to the environment. Are we going to be able to perceive social cues? Are in the case of marine mammals, are they capable of using sonar the way they had before? All of these kind of things I think are plausible, and we've got solid evidence in other kind of cases that give us insight about this. But we also have to recognize that heavy metals that they're exposed to are going to be interacting with that, as well as, you can imagine, perfluorinated chemicals, et cetera, et cetera. It just goes on and on. It's shocking to me that that there are countries that are focused on recycling plastic rather than reducing them, because the health effects, the health consequences of microplastics, of plastics in the environment, of plastic food containers, et cetera, is significant. We can't ignore this. And we're seeing these kind of problems throughout the animal kingdom. So I think that it's you know, it's naive to think that we're somehow above that.
Mm-hmm. Well, thank you for that response. I completely agree. Given that PCBs are still out there in the environment and in soil, do you see any way of launching some kind of cleanup effort?
Boy,
Is that even possible?
it's a good question. There was that, you know, there was significant PCB contamination in Pittsfield, Massachusetts. And of course, they dredged it, which then released a lot of those PCBs into the Housatonic. How do we deal with this? You know, it's, yeah. I'm not a reclamation kind of person, but we have to do better, and we can't continue to ignore this. One of the problems I guess that I have felt is that chemical effects on IQ don't alter how cute a baby is. And as long as we can imagine that they're cute and fine, we're not going to do anything about it. The case of thalidomide might inform us both about how the timing of exposure during development is important, but also warn us that we don't need to see a tragic physical anomaly to have tragic consequences. Mm-hmm. And that's a little bit long-winded, but it worries me a lot, because people don't pay attention to IQ. So what if you lose 5 to 6 points? I was at a meeting once where a physician said that children with congenital hypothyroidism who are treated appropriately as a population have about a 6-point deficit in IQ, which is fine. And there were people in the audience that rejected that, because 6 points on a population level is affecting both sides of that bell curve. This is a huge problem that I think is being ignored because we don't see obvious issues.
Well, I think what's also troubling is the idea that you mentioned, that interaction in society, or being able to function in a community is threatened, basically. That perhaps some of the things that are happening now in the world are partially triggered by people's incapacity to relate to each other in a way that they have 50 years ago, whether we didn't have PCBs altering how we think or how we relate to each other.
Yeah.
Yeah. So what's the next step for all of this, given the different mechanisms that can happen with PCB exposures and the many different sources of exposure? What are the next steps that we can protect ourselves from this? Or do we start screening people for levels of PCB in their bodies and therefore do some kind of treatment, not necessarily treatment, but helping people acknowledge how their brains not be functioning as perhaps they could, and doing some kind of remediation activities with humans? What are the next steps for all of this?
Well, I think there's a couple approaches. One is, I'd at least like to think that the body of our work did help the European Commission decide to fund the Urian clusters. So there are 8 large projects that are focused on finding new ways to identify chemical hazards. Now, two of those projects are focused specifically on thyroid hormone. I'm part of the Athena project that is trying to identify both in vitro and in vivo endpoints that will give us a better picture of chemical effects on thyroid hormone system disruption. So I think we need to get a lot better. I mean, up until this time, a reduction in thyroid hormone levels in rodents was pretty much the gold standard. It was measure T4 in the blood, measure thyroid histopathology, and that's it. And that's totally inadequate. So I do think that there is a recognition among risk assessors that we can't stop with blood levels of hormone. We have to look at specific kind of endpoints in the developing brain to give us better insight. In terms of how we can manage this, you know, I have worked with the Endocrine Society for quite a while, and there is kind of discussion among physicians about how to better advise patients with respect to these kind of issues, not just PCBs, but a kind of battery of chemicals that they may be exposed to. One argument is that, of course, physicians will always recommend to their patients not to smoke, not to, there's a series of things that we know increase your risk of disease. But we also know that there are chemicals that increase the risk of disease and how you can avoid them. Until we have regulations that are effective and the political will to protect people, I do think that the best we can do is try to figure out how to avoid chemical exposures. I'm pretty shocked when I find people, young people, especially young people with young children or that are attempting to get pregnant, who put plastic in their microwave. I mean, we still need to teach people how to have a lower risk profile when it comes to chemical exposures.
We really do. Now, we have a battery of really good questions here that have just appeared, so I'm going to read these out to you. A few studies have suggested in the past that dioxins may increase blood levels of thyroid hormone. These have tended to be in men. You are postulating a two-step process by which increased CYP1A1 leads to hydroxylated PCBs, which then increase peripheral thyroid hormone. Is there any reason to think that this could relate to sex differences in PCB effects? And if so, how?
Yeah, I think one kind of clarification there is that this two-step process was not related to thyroid hormone levels, because in the rodents that we studied, I don't think we can say this for humans, but PCBs caused a reduction in thyroid hormone levels in the blood. But at the same time, we saw an activation of thyroid hormone receptor in liver and heart, pituitary, brain. I didn't show a lot of that data. And we showed in cell lines that this was related to the CYP1A1 effect on non-dioxin-like congeners. So that's kind of a clarification. What was the second part of that question?
Oh, whether there were sex differences or...
Oh, sex differences. I mean, it's entirely possible. There's, men and women do have different metabolic profiles, and so that could easily be part of that. We actually haven't seen sex differences in the effect of PCBs on thyroid hormone receptor activation or thyroid hormone system. It certainly doesn't mean that it doesn't occur for other endpoints, especially in humans, but metabolism is certainly plausible for sure.
Okay. Someone else, thank you so much for this talk and all your powerful work. Two quick questions. Do you think other modifiers of thyroid physiology are masking PCB effects in most of our science? For example, chemicals from cigarettes can disrupt thyroid physiology.
Yeah, well, I mean, I think cigarettes are kind of a good example because people volunteer to smoke, and we can capture that in an epidemiological study. And of course, cigarettes have not only cadmium, as you're saying, but thiocyanate, which affects iodine uptake into the thyroid gland. We know that people who are exposed to perchlorate, and actually we're all exposed, but women exposed to perchlorate are more sensitive to the thyroid hormone reduction effect of perchlorate if they smoke, largely because of thiocyanate. But we also know that the sodium iodide symporter, is sensitive to bromine, to fluoride, to a lot of these negative ions, and this is always going to be a case. And so in epidemiological studies, oftentimes, I think the effect of a chemical on some output like thyroid hormone could very easily be masked by the variability, both the intra-individual variability in thyroid hormone itself, but also the difference in exposures to chemicals like dioxins and different lifestyle.
Mm-hmm. Mm-hmm. The same person has a second question. Are you aware of any auditory epidemiology related to the finding you described?
It's a good question. So the kind of audiology that has been done both for PCBs as well as for thyroid hormone has more to do with hearing, not so much perception. So I'm not sure how they would approach that. There are some probably EEG. There are some auditory evoked potential studies that are done in humans. I'm not that familiar with it, and I'm not exactly sure how a disorganized auditory cortex would reveal itself in some endpoint. I'm sure there are other people who have a very clear perception of what to predict there, but that's not me.
Okay. Have children that were born in mothers exposed to PCBs, have they been assessed as a group for neurological deficits in communities like, for example, Anniston, Alabama, or people that are eating a lot of fish from the Hudson River and other sources?
Both of those populations have been evaluated for that, yes.
Okay. Mm-hmm. What was the result of those studies?
So, yeah, there's, the kind of endpoints that they've looked at over the years has changed. I think one of the very first studies was done in Michigan, and they were looking just at IQ. IQ is kind of a blunt instrument, I think, and it's not that specific, but there have been a number of domains that have been evaluated, impulse control, et cetera, that have been looked at. The review by Shantz and Rice, I think, is a really good one to give you a good idea of those studies.
Okay, that's good to know. What type, 3 things would you recommend to a woman who's just found she's pregnant? How would you word the connection between IQ and exposures without frightening the pregnant woman?
Right. So what in fact, I have 2 grandchildren from 2 different young women that I kind of said the same thing to, which is, first of all, make sure you have good prenatal vitamins that includes iodine. And then stay away from certain foods. There's a lot of information about what kind of foods to stay away from as a pregnant woman, but also the habits you use putting plastic in the microwave or even the dishwasher, the kinds of plastics that you use, the way we store food, also ultra processed foods. Let's cook. So if you focus on the things they can do, you know that you're not going to eliminate exposures. We don't have the capacity to do that, but we do have the capacity to reduce some of the kind of exposures that come based on the decisions we make about how we make our food and where we go. I guess for me, fortunately, both women live in a relatively healthy place. They're not next to a big agricultural field or next to a factory, et cetera. We don't always have choices about where we live. And that's, those are things that we may not be able to do anything about. But as long as we focus on what we can do, I think it gives them power. It's more enabling than paralyzing.
Very good answer. Thank you for that. Well, we reached the end of the time allotted, but is there anything that you feel that you'd like to ask yourself, or you have a question for yourself that perhaps there's something you haven't quite covered that you wanted to at this point?
I think I have, just to reinforce that that I think the regulatory system was kind of, it developed focusing on death and disease, things that are very obvious. Where we are now, and especially I think with population, non-communicable diseases, et cetera, we're looking at a slow death and an erosion of functionality and quality of life that we should be able to address. We've got the science to do it. We've got the information to do it. We need to have, the legislative and regulatory mechanism to do that, because we can't protect ourselves from chemicals like PFAS that are everywhere, and they just continue to accumulate.
They certainly do. They certainly do. Well, thank you very much. This has been an excellent presentation.
Thank you.
I think everyone has really appreciated your work. We've gotten a lot of comments coming in, just thanking you for the work you're doing and bringing it out to the public. So it's been an honor to talk with you today, and I think it'll be that we'll be checking in with you later on for more conversations, because this topic is not going to go anyway soon, and we need to keep working on it, keep raising it to the population and to regulators and to people that test chemicals for toxicity and using the wrong methodologies quite often, as you mentioned, in terms of NAMs.
Yeah.
Okay, so thanks a lot, Tom. Really appreciate it.
Okay, thank you.
Let's go back now to Rachel to close us out. So thanks again.
Great. Thank you so much, Charles, and thank you very much, Tom, for this really important research and for taking the time to talk with us today. And thanks to everyone who put excellent questions and comments into the chat and the Q&A box. We really appreciate the work that all of you are doing as well. So we'll just wrap up with a few quick announcements. A video recording will be available on Che's website soon, and you'll all receive an email with a link to the video. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on Che's Partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to Che and/or any of these partners. We also invite you to join us for upcoming webinars. The next EDC Strategies Partnership webinar will be on January 9th, and the topic will be Food Contact Materials, a roadmap to improved safety testing. And we'll put that link into the chat in just a moment. And with that, we'd just like to thank you again very much for joining us today. Thanks so much, Tom, for your research and for your excellent presentation. And thanks to Kristen Schaefer and to Sarah Howard for their support behind the scenes. Thanks again to all of you for joining us.